do this. So, one, I was saying, growing up with and seeing this sort of tension, and then continuing to see this this sort of rift between allopathic and non-allopathic medicine is somewhat eternally frustrating. But I think it certainly goes in both directions. The physicians, allopathic physicians, are oftentimes not so keen on sending their patients to non-allopathic physicians, and I think the other way around. I think there's a lot of skepticism about Western medicine and allopathic medicine from the alternative medicine crowd, or whatever words are used. I'm not in with the lingo these days, but because of that, I really like to at least try to bridge it in some regard. And so part of what I want to talk about is basic biology at the sort of molecular and cellular level, and also be a resource or be somebody here who has a very different view on what is medicine or what is the body than what your view is, or some of yours. And hopefully there's a lot of intersections there that already exist. So that's sort of the big thing. So I'll talk a little bit about cell bio, and hopefully I think it's particularly important, probably more so. I look at a normal allopathic physician who went through medical school, and we spend a tremendous amount of energy learning this crap, which from a normal sort of Western medicine point of view, we don't use it at all, because we almost never touch a patient. I mean, it's kind of a crazy thing. And I would say the Western medicine view, take away surgeons, but the internal medicine view of what is the body is essentially treating us like a bag of blood and chemicals, and we try to manipulate that. So we don't really look at the cell structure, but in your line of work, you absolutely look at the cell structure. You touch it. You use pressure as your mode of healing, and so I think it's a particularly important aspect of what you. Or hopefully at least will be an aid or another tool to visualize, to really see how complex and amazing and beautiful every cell is in our body, because that's essentially what we all work on. And then I'll talk a little bit about, well, if there's interest, I like to talk a little bit about vaccines and antibiotics and the differences. Part of that is because, so I started saying before, you're all healthcare practitioners, and patients will come to you for advice. And I've seen a tremendous amount of misinformation given out about what is a vaccine, what is an antibiotic, and there's just, and I think that's a huge component of healing, is both prevention and obviously treatment, and those kind of encompass both. So hopefully we'll get to that, and I want that in particular to really be a discussion. I want all this to be a discussion. I just like to show pretty pictures and then talk. All right, so we're going to start with, I'm just going to play this video through, and then I'm going to play it through again, but we're going to discuss it as it goes along. And so the majority of this video, this is a beautiful animation that was put together by a group at Harvard. They do a lot of really wonderful animations now. And all of the elements that you'll see in this video are actually taken from the molecular biology of a microscopy. So it's, even if things look like just strange conformations, it's actually very detailed and specific about what a lot of these elements look like. So in that, we're starting out in a blood vessel, and then very quickly we're going to go into the inside of a cell. And so I just want you to, and you should recognize it, but when we go inside the cell, I'll let you know so that you can start to appreciate that in that way. And then afterwards we'll go through. So here, this is a blood vessel. And this is a lymphocyte, or a white blood cell, an immune cell. And that's what we'll spend the majority of the time inside of. About 10 trillion bacteria. So and each of the bacteria have very similar mechanisms, and all of the components of that cell that you just saw are pretty consistent. Those are sort of the basics that are involved with almost every cell in our body in some form or another. So it's pretty amazing to consider how many cells we have. So a trillion is a hard number to comprehend, but if you consider that you can fit somewhere around 1 million of them on a pen tip and things like that, it's pretty exciting.
Is that inside a white blood cell?
That was all inside of a white blood cell, yeah. So this is a leukocyte, so they're all the immune cells that help us defend ourselves against infections.
The one that was roaming?
Sorry?
The one that was roaming? Like that?
Yeah, the rolling cells.
Yeah.
So that's essentially what they do. If you get injured or a cut or some sort of inflammation on your finger, when you break through the skin cells, they start to, or you get an infection, or your body just detects some invader. There start to be chemicals that are sent out, and there are a lot of times like the chemicals are little proteins, and they look like those little nuggets that seem to be floating all around. They're called cytokines or chemokines, and they essentially send out this message just through chemical gradients. And so you have a lesion or some sort of laceration or something. Those cells start sending out lots of chemicals that they're making through just processing their DNA. And ultimately, these white blood cells start to follow that chemical trail, and they get attracted just based on chemical gradients. And then they ultimately can squish down into little pancakes and get through your blood vessels,
They
just
go,
like
the,
what we
they
showed
go
last
toward
time.
the density of the chemical, they go
Mhm.
into the density.
Yeah.
That's
So it's pretty amazing. I mean, inflammation is a rapid and incredible thing. So, let's see.
I have a question before you
sure.
start going through the video again. So you said there's 1 trillion cells in the body, and there's 10 trillion bacteria. So this might sound like a silly question, but so are you saying the bacteria isn't cellular?
No, there's a trillion human cells.
Ah.
So each human cell has a full complement of human DNA and chromosomes.
Okay.
And so there's about 10 trillion bacteria of all different species, and they just sort of live in and amongst us. Which was kind of an interesting, I mean, from an evolutionary point of view, you all were talking a little bit about vertebrate evolution and the role of that. And what's quite interesting is that bacteria, maybe it's obvious or maybe it's not, evolve at extremely fast rates relative to humans. In general, the larger the species, the slower the evolution relative to a small species. But it kind of brings to question what is a human, since we have these tremendous numbers of bacteria that are far outnumbering the numbers of human cells that exist in us. And they're evolving at a much faster pace than we are, so it's kind of raises some interesting questions about what is a human species. Yeah.
Given what you had just seen, and then you said inflammation is a very interesting thing. Can you just do like a quick brief on what inflammation is from what we just saw?
Oh, you're going to go
Sure.
through the video again, right? And
Yeah, we'll go
talk
through
about
that again. However, none of that has anything to do with inflammation. I think inflammation is more at a multicellular level. So we were looking within the cell, but all of what we were seeing is essentially the cell doing its thing. It's whether that's making proteins or moving or rolling, all of it needs to be done through the production of proteins. So proteins are the building blocks of cells. Without them, nothing happens, essentially. And so that cell, they were essentially showing a cell getting ready to go and help out with some inflammatory process. So whether there were a number of times, and I will point it out, when the cell's making proteins and putting them and packaging them in vesicles to ship out, kind of like a FedEx or UPS truck is like shipping out all these cytokines and chemokines to alert more cells, and those are also used to detect things. And so at the single cellular level, inflammation doesn't really exist. but it's really a multicellular process. Let's see. So my specialty is more in immunology, infectious disease, and math. However, I have a lot of background in sort of cell bio because that is sort of what allows inflammation and infectious disease processes to occur, which is why I like this video, because it kind of brings everything together. So I'm going to show it again, and absolutely stop if you have any questions that I'm not talking about. I'm just going to point things out and allow the video to sort of move us through this conversation a bit. But I hope that you will attempt to think about what this means. I think it's very easy to look at us and think that we're very fluid, and we are. All of this is living, or not living. I mean, inside of a cell, it's hard to say that that's life, even though a lot of times it looks like life. But that's a very philosophical question. Well, I think if you take a single cell in a human eukaryote and you take a protein in that cell, the proteins, I would absolutely not consider a protein living, but I would consider a whole cell living. So it's at what point does a protein and life start? I don't know what the answer to that is, but I know that the fact that I can take two chemicals and make a bunch of proteins in a lab, to me, I don't think that I'm creating life there. But life is made up of a bunch of inanimate things that work together. So, but that we'd be more than happy to talk about
And they're
that.
synthetic, huh? They're
What's that?
Two synthetic proteins?
Yeah, I mean
Creating-
we can make synthetic proteins very easily now, and we just make them, and you know, and so I don't believe that proteins encapsulate what is life, but they. It would be like saying that if you took a little bit of plasma from your blood or something like that, I wouldn't call that life either. It has no cells or no anything in it. So it's interesting where the breakdown occurs.
So what did you start to say about the fluid?
At which point? So what I find so amazing about this is in any of these cells, so our bodies are made up of cells, obviously, and cells, I would say, we were talking about this a little bit last night, what's the distinction between fluids? So CSF, or cerebrospinal fluid, is usually acellular, doesn't have cells unless you have meningitis or something along those lines, which hopefully that doesn't last too long. So you have this this fluid that's sort of crystal structures, whether that's salts or sugars, but there's usually not any cells. Whereas tissue or blood, so blood is primarily plasma, which isn't cells, but then it's filled with red blood cells and white blood cells and debris from other cells that are sloughing off and things like that. And the body feels fluid, and we throw out terms like it's 90% water or 80% water, and that's true, but within all of that are these amazing little cities. Like I look at every cell as though it's its own little city, and it pretty much has about that much activity. If you were to take a whole of New York City, you could fit that kind of movement and take a sort of top-down view of Manhattan. You have as many little moving parts in one cell as you do in an entire city, which I just find incredible. And so, to me, it starts to, from a visualization point of view, what is a fluid inside the body, and what is just like, you would never call Manhattan a fluid, but you could also look at it as such. It's got these amazing little pathways that move through it, and in a very fluid-like fashion, because things tend to go towards efficiency, or I can't say that Manhattan anyway, but
Certainly not Boston.
but they work at it.
Yeah, Boston, definitely.
Yes, certainly Boston.
So I think the point is just that that what we think of as a fluid is so much more complex when it comes to living creatures and what's happening in each of our cells. And so we can see that here. So this is a blood vessel, and we just see blood just pouring through, think about how quickly blood so now we're talking about at the organismal level, not at the cellular level. Our heart pumps blood through our whole body and through a tremendous number of capillaries, just at amazing rates. And I get so excited. Talking about capillaries is such a
The
cool
blood
thing.
vessel is really not smooth on the inside.
No, blood
like
vessel-
crevices.
Exactly. So it's not smooth, and that's why you get plaque buildup and heart attacks and things like that. Because if you start getting little ridges between cells that a little bit of cholesterol builds up on. There's a guy named William Hurst. He was one of the founders of what we now know of. He was one of the original sort of explorers of the blood vessels in the heart at a microscopic level. He was actually down at Emory. He just died a couple of years ago. But I loved hearing him talk because he'd say before you're even born, you start getting white streaks in your blood vessels and things like that. And that's essentially even before you're born, you start building up cholesterol and you're on your way to heart attacks. Essentially, which is interesting. But no, it's not smooth, and it's actually made up. The blood vessel you see here is made up of endothelial cells. So everything in our body, every piece of tissue that you see, whether it's a blood vessel, fingernail's different. But any bit of tissue that you see is made up of little cells, and they come together and they stick together like Velcro, and there's little junctions between them all, and they just all happen to work together. Not just happen, but there's a long evolutionary chain there.
I have a question.
Yeah.
So does the cholesterol serve a purpose in perhaps smoothing it out for easier transportation of the rest of what's going through there, and its body's, it can't maintain the homeostasis to make sure that it stays at a certain level so it does build up to overproduction? Does it serve an actual purpose by building up? Because if it's there, is it just to make things easier to get through those areas without getting stuck on those little indents?
I would probably say that it doesn't serve that purpose because we don't see it completely covering all the blood vessels by any means. We just see it building up like at branches and things like that.
Where it bifurcates.
Yeah. And so cholesterol has extremely important uses when it comes to the actual components of the cell and how the cell functions and our mitochondria and respiration. From a blood vessel, I would say that there was no real need to, or evolution never got rid of cholesterol buildup in blood vessels because until recently, no organisms lived to have heart attacks. Now with modern medicine, we're living long enough where that's becoming a limitation. But prior to that, it didn't hurt the species enough. By the time you have your kids, or by the time you have a heart attack, you've already had your kids, and that's sort of what allows evolution. Doesn't matter once you have your children, what you're doing really from an evolutionary point of view. Your genes are already out there again. So I would say it's a really interesting, who knows from an evolutionary point of view if there is some utility, but I think from a purely physical sort of viscosity.
I was just trying to come up with some sort of, example. I didn't know if it served another purpose.
Right. But not that I know of. But who knows? There's a tremendous amount that science doesn't know yet. So in any case, we're the blood vessels. You see all these red blood vessels and platelets coming down, and then essentially what is not being depicted here is the plasma. So blood is red because of the red blood vessels. But actually everything else is a fluid inside of here, and it's not being shown, but that's what the clear stuff is here. And then these guys are rolling along on the blood vessel, and as you mentioned before, they're sort of waiting to get into the endothelial cells. So the blood vessel is made up of just a tremendous number of cells, and they can kind of squeeze through. I'll let it go. So here, this is actually how that vessel is, that blood cell is rolling along. It actually works with this amazing turnover of Velcro, essentially. And it's not truly Velcro with hooks, but it's magnetic. I like to think of every single itty-bitty, to the smallest little atom and quark in our body. Everything is just binomial. It's just yes, no. The reason that things do anything is because there's electric charge, and positive likes to go to negative, and negative doesn't like to go to negative. And so that's how these are working here. Everything that you see here is actually working based on some just principle of positive and negative, and then they kind of fit together. So a positive charge here likes to go to a negative one here. And then in the case of proteins, they have these amazing folds that all sort of work together, and they'll fold, and then they fold in such a way, in this case, where they can then release very quickly. As soon as they come together, they sort of change again, and that causes them to release. And so you see this extremely high turnover of these little hair-like structures. They're proteins. And again, almost everything you'll see in this, and I don't believe they even show DNA. They do show mRNA. They show RNA at one point. So besides that, everything more or less that you see in this video
I thought I
is
saw
a
the doing
protein.
the strands. I thought it was doing that. The
Yeah, so that's
strands.
RNA.
That's
Yeah.
RNA.
Yeah, the DNA stays in its chromosomes inside. And
Here it looks like there was protein, and then there was another protein that was the severin protein.
That was actually an actin.
Okay.
It was, so that was a severin protein cutting another protein.
Right.
So everything, these are all proteins, and they're all amino acids. So this might be made up of, 1,000 amino acids right here. And amino acids are sort of, that's what the DNA codes for. It codes for chains of amino acids, which then once they're made, and I'll show you the ribosome that's making them, they just fold into these little molecular machines that have no real, on their own they don't move at all, but as soon as they come into contact with another charged thing, they start to change shape, and then they, in this case, grab onto each other. So the cell is literally rolling by pushing itself along with Velcro, or with these little pseudo Velcro things. Where did the mouse go? So then you can kind of see them adhering there. They're just being attracted to it, which is what's pulling the cell membrane up. So here that would be a junction between two cells that they're showing here. So there are these concentrations of selectins and all these different proteins which allow, for endothelial cells, they bind very tightly. And in particular, there's another group of endothelial cells that are extremely tightly bound, which would be the ones, they're the most tightly bound in the body, more or less, as a whole structure, and those are the ones that surround the cerebral spinal fluid. And that's because we don't like things to leak into our brains, essentially. So they hold extremely, extremely tightly, and it also prevents those white blood cells from getting through. So whereas an endothelial vessel, like a blood vessel, allows white blood cells to get through, and it has a little bit of play in terms of how tightly the cells are being held together. But you all work with the cerebral spinal fluid, at least in this course, and they are held extremely tight. You can kind of think of them as like plastic. They don't let anything through, or they don't want to let anything through, except for some salts and things that can just sort of float right through the cells themselves. It's also why when we get a bacterial infection, it tends to have very high fatality rates because our immune system isn't privileged to go in there. So if a bacteria makes its way in there, or if a virus makes its way in there, it just kind of has a field day without, and white blood cells can't really get in until the membrane has become so inflamed and affected by the pathogen that all of a sudden our blood cells can actually enter in a worthwhile number.
So where specifically are the endothelial cells? Are you saying that the meninges is endothelia?
Yeah.
Isn't it a single layer?
It might be epithelial. I forget like what. It might be. I mean, they're the same types of cells. I forget what the actual. We never really talk about.
The walls
Whether
of
it's
the blood
endothelial or
vessels are endothelial.
Yeah, and so I'm not actually sure what the walls of the CSF, like what the walls of the vesicles and or ventricles and such are, but I think they're a type of endothelial cell.
Is it a single layer?
Yeah. And they hold extremely, extremely tightly. So that's the main point there. I forget what the actual name is, but they have these what are called tight junctions, which hold a lot of tissue together, and the meninges just have a tremendous number of tight junctions. So this you could think of as a junction between two cells where it's like a high concentration of these proteins, and it kind of if you look at a microscope image of it, it actually looks like if you had two pieces of fabric or a strap or something sewn together, and it just looks like the thread very tightly woven to hold two pieces of fabric together, and that's sort of what holds our vessels together. This is what our cells are all made out of. So here, these are our lipids. So every cell is sort of encompassed or encapsulated by a membrane, and the membrane is made out of fat. And what happens when we put water and oil together? They separate. Yeah. So which is exactly why our cells, if you take a drop of oil and look at it under a microscope, it's going to form a sphere very rapidly in water, because that's the most efficient way that it can sort of survive and get to the surface of the water, if you will. And our cells do exactly the same thing. They're actually made up of all these lipids, but what you're seeing here at the top right now is you have lipids which are just fat molecules. They're carbons. They're just carbon chains put together. But on the ends you have what's called hydrophilic ends and then hydrophobic centers. So the hydrophobic centers means they don't like water. The hydrophilic ends is that they do like water. So what a cell is made out of is it's got these water-loving ends, and the middle of it is these, and actually they show a good picture, so I wasn't discussing it. So these are lipid and cholesterol wraps here. I'll come back to that in a minute.
And they're on the outside of the cell right now?
We're still on the outside. So these things are actually these wraps help to, just like, they're called wraps for a reason, they just help transport stuff along the cell surface. But I want to get to a picture of this lipid bilayer. Here we go. So these are the hydrophilic ends of the fat chain, so these are triglycerides. And then in here we have the hydrophobic center. So what that does is it literally creates this wall around a cell that doesn't allow the water to go through. So if we think of there being water on either side, and the water is obviously filled with lots of different compounds and proteins and salts, so it's not pure water by any means, it's at the very least saline. This is essentially what stops our cells from just going crazy and sort of blowing up all the time and things like that. And it allows very, very selective transport of ions and proteins and things like that into and out of the cell. So we have this sort of barrier. Without this barrier, we wouldn't survive. This is essentially integral to all life, at least all life that we've ever been able to see on Earth is bound by these sort of plasma membranes. And they're made, they're all made from cell division. It's just that there's a constant production of lipid being produced all the time. And you can see here what they're showing is cholesterol is being used, so cholesterol is integral to the cell surface. And then there's a lot of proteins that just sort of live. So this is a bisection, we're just sort of taking a cross-section of this membrane, but there's a lot of transmembrane proteins, they're called, that just kind of go through each end. And they get anchored by this hydrophobic area. So this would be a hydrophobic part of this protein, and it likes to live here. So once it gets anchored in there, this is hydrophilic, that's hydrophilic. Once it gets anchored in there, it's not going to move up or down, because this part doesn't want to see this part. And so it just kind of gets anchored into the membrane.
And then what do? They share information and resources
Yeah,
and stuff?
exactly. So we could think of this, and that's not what this one is trying to depict, but they all kind of look like these blobs. The way that cells communicate, I'm sure that they showed it sometime, I forgot. But the way cells communicate, I can do this is awesome. There's what are called, we have all different types of receptors that live on the surface, and they just kind of like chill out on the surface. And their transmembrane receptors are the ones that, if this were a receptor here, I might I might be a signal, say I'm a chemokine and I want to tell this cell to do something, whether it's to start rolling that way. We can say, I might go and I might grab on like that, and it will be like a perfect conformation, and I'll grab on and it's just based again on like magnetic charges at the atomic level. But here it's also based on conformation. And what this will do is, it's not gonna drag it, because I might just be a floating chemokine, I might just a chemical signal. But it will be enough for me to link onto this, and what it does is it sends sort of I don't want to say a message, it's not like a neuron sending an electrical message, but it's more, it just changes the shape. It's sort of like a ripple effect. You grab on and it starts to change the shape, and it's all based on energy. It's based on, now that I have this other thing attached to me, how can I change my shape to get to the lowest energy state? And then we're talking at an atomic level now, or molecular. So it changes shape in very subtle ways, and that message, when it actually changes the shape, it gets passed straight through here to the intracellular part of this receptor. And then this ends up being changed, just a slight amount. This will like change shape, whether it curls up a little bit or whether it opens out. And that enables something else to then attach to it or detach from it. And that's essentially how a signal gets propagated from touching from the outside of the cell. It grabs onto this receptor and it moves the signal right through to the inside of the cell. So at a molecular level, that's essentially how all of our cells are signaling to each other, which is kind of cool. Because if you think about it, at this level, physical force is making very little difference. There's not, if we press on a cell to an extent that there are some signals that get passed through with physical force, but a large majority of the cells are actually, physical force might be altering some chemicals that end up altering the actual chemical signals that are put through. So that's why if I hit myself, that will cause a chemical signature to cause inflammation there. It's not going to be, my blood vessels don't feel these ripples of sort of forces. But we know from embryology, for example, that it's not purely chemical, because that's a very clear physical fluid dynamic sort of function there. And we can also see, a lot of times we see, I'm trying to remember the phrase that we use, a sea of, I can't recall at the moment, but essentially none of this stuff is static. So even these lipids that are making up the cell wall are just constantly moving, and they're constantly, just like if you think of water, just sort of the water molecules, or unless they're ice, but they're always moving around each other, just constantly moving. The same thing here, these lipids are always moving and they're always sort of flowing, and that allows a cell to remain extremely dynamic. So this is what I was discussing before, a chemokine. So this is how signaling occurs, and if I get an infection or my heart rate wants to increase, I send out chemical signals to tell my whole body to sort of follow through with whatever signaling I want to do. So we have both chemical and electrical signals in our bodies. This always makes me think of dinosaurs for some reason.
Yes.
So now we're inside of a cell. So you can see this lipid raft, the underside is sort of carrying, they're used to transport these transmembrane proteins. Because once a protein is embedded in the cell membrane on its own, without a lipid raft, it has a hard time moving. It can't, because all these lipids are just kind of moving around it randomly, and so it just stays. So essentially the lipid rafts are used to actually transport stuff. And again, they're moving based on some chemical signature. And so they're the ones, they're kind of like icebreakers, like on a boat. They're able to just push everything out of the way and carry something to where it needs to be. When it drops it off, it just sort of stays then anchored in the membrane.
So at the top is the membrane of the cell.
Yeah, so now we're kind of like inside looking up into it. This is one of the G protein receptors. This is one of the basic components of communication in the whole, in any organism. They're these really cool, they're transmembrane ones. they might change something, and it might end up, instead of taking a signal from the outside in, the G protein, once it gets that signal from the outside in, it might then sort of make its way over. It might stretch out a big arm and touch something like this, which is just an intracellular protein. It doesn't have an extracellular component. And then that might end up, it's all about cascades and sort of chains of events. So it might touch this one and tell that one to do something. And you'll see all of these sort of thread-like structures, those are called actin, and we'll get back to those, or these might be microtubules. Yeah. So spectrin is made out of it's called, well, it's a type of tubule, and there's a few major components. There's actin, and then sort of what we just call microtubules as a catch-all. But they're sort of like, and you can see it here. They're the structure of the cell. So the cell is made out of these membranes, but then inside of it they tend to have a scaffolding, and so that's what these are, essentially the scaffolding. And it looks exactly like we would think of what we would hope to do if we were building a skeleton of a sphere.
Does that scaffolding have a different look in different types of cells?
Oh yeah, it has extremely, so that's one of the major things that will allow different cells to have different shapes. They'll always form, like spectrum will always form these tetramers, but whether or not they go in the whole sphere or they are in different, it's more about the density of how many there are. So certain cells that are very, very mobile and very, very plastic and like to flatten down a lot, they might have fewer of these, because these are sort of stiffer and allow a cell to stay more globular. So this is all actin. Actin is sort of the spine or the skeleton of a cell, and it goes all through the whole thing. So we have a circle, we have these spectrin filaments. They're very thin filaments that help just support it, and there's also there's other uses for it, which I think this video will depict. And then actin is sort of like the internal structures. It's just it tends to form these really nice, very organized webs that tend to be at orthogonal or 90-degree angles to each other, and they really support the cell's whole integrity. So the amazing thing is all of this stuff, humans like to think we're efficient, but our cellular structures are much more efficient than we are. So you can see how many filaments there are, and that's essentially what's enabling. Not only are they creating structure, but they also, which you'll see with the little thing that walks, they're the roadways as well. They enable transport within the cell. But unlike humans and the infrastructure we build, these things can be built up and removed extremely rapidly, which is just, I think, one of the most amazing aspects of cell biology. So here this is showing them being built up. These are actin filaments, and again, they just come together based purely on charge and being the right fit. A lot of times we use the analogy of a lock and key. So if they're the right charge, that brings them together in the first place to attract each other. And then they have to be the right fit. So that's why you don't end up seeing a microtubule protein or a G protein binding with an actin protein. They're two totally different fits, and they might be at two totally different scales in terms of size. But it's the charge that brings them sort of close to each other, and in a cell, everything is always, there's so many little protein bits moving around that they're bound to come in contact, essentially. And so in particular, actin is at very high concentration. So it's able to do this thing where once it wants to build, or wants, meaning the density of the little particles gets high enough that all of a sudden there's just sort of this spontaneous occurrence where it starts building up. And once it does that, it attracts more and more, just like we're seeing here, and they just form into these big long actin filaments. And it's kind of like vertebrae in a way, in that well, all of our vertebrae are different, T4 and T5 and all those, but if you think of them as all being the exact same, if you thought of a skeleton as, or the spine as being made up of the exact same little bits, it's sort of like that. These are all actin proteins are clones of each other. They're just little molecular rocks, essentially, that end up being able to be stacked on top of each other very nicely like this. And then this is a protein, which it's an enzyme, which enzyme is just a protein that does something. So an actin protein is not an enzyme because it doesn't have an actual sort of molecular machine aspect to it. It's just sort of like the difference between a brick versus a drill. Neither is particularly animate, but one can actually perform a function besides just being structural. So the severin protein is a type of enzyme, and its whole goal in life is to just cut actin, in this case. Once the actin gets cut, so it gets built up into these long things, and then once it gets cut, it becomes destabilized, and you end up seeing it just fall apart again. And that's how when a cell is moving like we saw, it needs to constantly be changing its structure all the time in order to move. Microtubules are a very similar thing, but they're more of like the highways and the real sort of like ropes of a cell. They enable things to be pulled together and moved on and things like that. And it's quite cool, and with an atomic microscope or just a light microscope, a very high-powered one, you can see all these. There are 9 different spindles that are making up each microtubule.
What passes through them?
So nothing passes through them, but things pass on them. Actually, what they're not showing in this video is there's actually 2 cores inside of it as well. So there's 9 little tubular filaments that make up the outside, and then there's 2, there's 7 that are on the outside, and then there's 2 on the inside, just for extra strength, essentially. It's very efficient. So here, this is probably my favorite part of this whole video. And part of it's because if you can actually see this stuff. I mean, it's like, you can't make this stuff up. It's just crazy. These little guys, they actually walk around, and they're doing it again, I would say, even though it looks like it has life, right? It looks like it's walking. And I think they show a better video of it, yeah. It's based purely on, it's not choosing to walk, it's not choosing to take each step. Essentially what happens, and biology was way ahead of us in terms of how it uses fuel. This is not showing ATP, so adenosine triphosphate, which is, ATP is truly the gasoline of our bodies. It enables everything to run, and that's why the only reason we breathe the oxygen is to enable us to make ATP, which is a whole nother really cool process. But essentially what happens is this little guy, these feet, are little combustion engines. And every time a foot goes down, it gets locked there. It can't move again until a new ATP comes in. So ATP is just a little molecule, and it's got 3 phosphates on it. And phosphates are really high, if you take one phosphate, it's an atom. It's one of the elements on the periodic table. So if you remember way back, periodic table, phosphate, phosphate is actually multiple elements, but the main thing is one part of it. And so 2 phosphates will actually come together, and there will be a very high energy bond between them. And just like in combustion, when you break a high energy bond, it gives off that energy. It's not sort of, it takes a little bit of wrapping your head around, because you'd think that it would require energy to break that bond, because you think of blowing stuff up kind of takes energy, but actually when the bond gets broken, it gives out energy. So that's how we produce energy. That's how combustion does it as well. So essentially an ATP will come in here. It's got 3 phosphates. That's the triphosphate part of ATP. And inside of here is a little combustion engine sort of thing that its whole purpose is just to break apart the phosphates. So it breaks 2 phosphates off, and essentially what that does is it gives it power, or it enables it to move. So it's locked until an ATP comes in. It cleaves the phosphates. The 2 phosphates go off this way. The AMP, adenosine monophosphate, so now it's only 1, will go off this way. And then once that happens, this thing can pick back up again, and then it immediately gets stuck down again because it's out of triphosphate. And so it's just a constant turnover of adenosine triphosphate turning into adenosine monophosphate, allowing this thing to just keep walking along on these little ridges. And again, it's all based on these interesting conformations that this little foot fits perfectly into this microtubule. So it's like little grooves on the road if you had, or like you could think of like a tank with its little, with its gears fitting into the I don't know what the words are for that. What's that?
The tread.
The tread, thank you, yeah.
And at the other end, what's
Yeah.
the other thing
Oh.
it's holding on to?
So this is, so these are its little hands essentially. This is a vesicle, and we'll see the production of these vesicles, but these are truly like the UPS service of our body. Vesicles are when we want to make a protein or we want to make anything. It usually happens from the nucleus. We look at our DNA, and our DNA says, oh, I want to make this protein or that protein. And then it goes from there and it makes RNA, and then into the ribosome makes the proteins. And then we'll show it in a minute, but then the proteins all get packaged into this vesicle. And then the vesicles will either just float to the surface of the cells, usually following some sort of actin filament, or they'll, if they need to be brought to a very specific place, these little microtubule highways will sort of allow the kinesins, is what this thing is called, the little foot thing, it's called the kinesin. They'll just kind of bring it along to where it needs to go. This is how neurons work. In fact, neurons need proteins to talk to each other. So at the junction of a neuron, do you guys talk much about neurons? I mean, I imagine you maybe do a little bit.
Yes, but we haven't done so yet.
Okay, so a neuron is super cool. I mean, it's just, you have two, if this is one arm of a neuron and this is another arm of a neuron, you have a junction, and it kind of looks like this. And so this neuron gets stimulated somehow, whether it's through touch, or whether it's through some other chemical, something or other, it gets stimulated, or just the heart maybe wants to send a signal to tell it to beat. And then this neuron needs to get the signal, but they're two different cells, and they're not touching. So what needs to happen is there's a chemical signal that connects this electrical signal to this one. And so when the electrical signal gets all the way up to here, this neuron lets out a whole bunch of chemicals, acetylcholine and all different things. And this one picks up those chemicals, and that chemical grating creates an electrical signal that passes it on. But what happens when this neuron starts to run out of chemicals to release? Because it releases them and it's firing quickly. This is also why your muscles get tired and things like that. Like you start to run out of chemicals. You'll need to replenish the chemicals here. So that replenishment of chemicals is actually quite slow, and it happens by these little things. They just so the electrical signal moves near to the speed of light, but then you have this really slow replenishment of chemicals in the head of this neuron, and it happens just by these little guys lumbering along, sort of waiting to get the chemicals there. So when you start exercising a lot, all of a sudden, or if you're just holding out, I mean, there's also the whole glucose problem of actually replenishing the nutrients, but also your nerves start to get depleted. You could also, if you just keep squeezing and pinching yourself for a long time, eventually it's not going to hurt anymore. And that's because your nerves can no longer fire because they're out of these chemicals, which I just think is cool. I don't know what happened. So that's essentially what these little traffickers are doing. These are the mitochondria. So this is where all the ATP is formed. And essentially all the oxygen we breathe in is the only reason we ever need oxygen. We don't even use the oxygen in a way. It's just kind of like this, it's a magnet essentially. The oxygen goes into the mitochondria, and this is actually Harvard has a whole other animation of just inside the mitochondria because it's just fascinating. But essentially it's all based on electric charges again. Oxygen is negatively charged. And so, or I should say it likes to be negatively charged. And when it gets into the mitochondria, electrons go towards it because it wants to have a negative charge. And the movement of electrons actually turns gears that end up bringing two phosphates together with AMP to make an ATP. And it does turn gears, which is just so cool. When the electrons they'll move through. It's kind of like it's like a hydroelectric dam or something, where the water is just turning a turbine and creating energy that way. Electrons moving towards oxygen inside of a mitochondria turn gears, and they actually cause an AMP to collide with two phosphates to create ATP. And so it's just inside of these gears in the
Where
mitochondria.
does the carbon come into power? I mean, carbon comes in there and attaches to the oxygen. At some point, it forms carbon dioxide.
It does. That happens. I believe that happens more outside of the mitochondria. Well, these form water. That process turns it into water, and then somewhere, I forget actually where it forms carbon dioxide. I'll have to think about that. I know that happens more at the respiratory level, though. Not inside the cell part. So these are them using the actin filaments as a guide instead of microtubules and being passed along. A lot of this stuff moves around in the cell just through like little wires and stuff to carry it along. So these we won't go into it, but these are super important for cell division. The centrosomes, that's sort of like, those will go out when a cell wants to divide. Each one of those will go to each side, and essentially they'll be responsible for pulling, once the nucleus falls apart, the DNA, each chromosome will end up getting pulled towards one centrosome. And then those will become sort of like, I'll show a picture of cell division, but that's essentially what ends up going to each side and pulling the chromosomes to each of their respective daughter cells. Okay, so now we're looking at a nucleus here, and essentially they don't ever go inside of the nucleus here, but DNA gets transcribed into mRNA, and that's based on this whole other awesome process with these tRNAs and the codes and stuff like that. But essentially what happens is you have DNA, it's bundled into its chromosomes, and to use that DNA we have to make it into something that's readable by what's called a ribosome to then create a protein. And that's the whole reason we have DNA, is to create proteins. Essentially it's to store the protein code. So inside of that all happens, the DNA turns into what's called messenger RNA inside of the nucleus, and then it gets sort of shot out of these pores that are in the nucleus. So the nucleus is sort of a protected little thing inside of our cells, and that's sort of, if you look at any picture of a cell, that's the main round thing that they usually show.
Is it in the mitochondria?
Sorry?
Isn't that in the mitochondria?
The
messenger cells?
No, the mitochondria is just these little electron transport chains with oxygen. So the nucleus here is sort of like a big ball in the middle of any cell, and then these messenger RNAs will actually be shot right out of the nucleus into what's called the cytoplasm of the cell. So there's the whole cell is here, then you have a nucleus, and in between, everything that we've been looking at has been happening in the cytoplasm. And so these messenger RNAs will get shot out of these pores, and they don't get shot out until they're sort of approved to be shot out. And that's this little cap that they're showing here. This is an adenosine cap, and they're just, it's the A's that make up DNA. Once they're sort of approved by the proofreaders, saying, okay, we have successfully and appropriately translated this piece of DNA, this gene, into an mRNA that's worthy of being made into a protein without having a mutation in it. Then it gets shot out of the pores, and this little A cap is what, the pores they're not just holes, they're actually little machines that will grab onto the A on the inside and sort of shoot it out. When I say A, I'm talking about their nucleotides or little molecules. What's that?
So the nucleus is not cytoplasm, it's made of DNA, or?
No, the nucleus is actually I showed you that lipid membrane that makes up the outside of a cell. There's another lipid membrane that makes up the nuclear sort of, you could think of it almost as a cell within a cell. But inside of the nucleus, you'll see all the chromosomes, and they just kind of fill the nucleus in a very dense fashion. But there's a lot of little things happening in the nucleus as well. That's how everything that happens is because chemical signals ultimately get in, and through the same sorts of processes that we've been looking at, cause different genes in the DNA to be transcribed into mRNA. So they get shot out, and then this is what you'll see here. They form these little rings, and then a ribosome will latch onto it, which they should show right here. And so what's happening is, right where it attached, every gene encodes a little transcription factor, they're called, but essentially there's little locations that all ribosomes will look for. So when ribosomes are just floating around, they look for these conserved sequences of RNA that are on all different genes have very similar conserved sequences, and that essentially just says, okay, I'm an RNA and I need a ribosome to attach to me here and read and produce a protein going from here down, so it doesn't end up like attaching here and just producing half of a leg. So the ribosome needs to know where on the mRNA strand to actually bind. So it always binds like right towards the top there, and that's based on just what that sequence is. And then it actually will just kind of go around and it reads it just like a computer might read code or anything else, or you might read a book. It will actually read it, and what it does is, in some sort of complicated fashion, which I won't go into, it ends up being able to read, they're called codons. And so if this is the mRNA here, these are made out of the nucleotides, like the bases that Watson and Crick showed the form of and things like that. But, they showed the form of DNA, but in any case, these are the bases. So these are single molecules, adenosine, or A, T, C, and G. And then every 3 of these, they're called codons. They actually, one codon will code for one codon, which is made up of 3 nucleotide bases, which your DNA is all just made of strings of bases, will code for 1 amino acid. So this is the protein that's being made, and so every amino acid, this might be a string of 100 amino acids by now. The ribosome literally has these little sensors, and it picks up the code. So it reads 3 at a time. It reads 3 bases and says, you're going to be this amino acid. And then it reads the next 3 bases. This is standing for this next amino acid. And it just brings in amino acids into its, so they're not showing it, but the amino acids float in on a different protein. They float into here, they bind, the amino acid goes, and then the little carrier protein leaves. And it just does that in this amazing, there's so many of them that it actually cycles through. So if I float into here but I'm not the right fit for those bases, I'll just be sort of kicked out without leaving my amino acid behind. And so it just sort of like is trial and error. But it happens, this is all happening, in milliseconds. It will make whole proteins and stuff. So this is how and even this ribosome here, well, it's a little different actually. Ribosomes are made out of protein. They're different, but but everything else is, and there's a good evolutionary reason why ribosomes can't be made out of protein. Can anyone think of why that would be?
So they don't get used up.
Well, that would be a good reason, but why else? If you go back to the beginning of life, why couldn't the thing that makes a protein itself be a protein?
Oh.
That's kind of the
Protein is
answer.
water.
What's that?
It just can't.
Right, because there are no proteins yet.
Right.
Right,
It's
so
making protein, so it
Exactly.
can't actually be a protein.
exactly. So ribosomes are actually this amazing. So this is why, from an evolutionary point of view, we actually believe that the first machines, the first bits of life, are actually made out of RNA. Because RNA is this really cool thing that can both store memory. It's like if you take DNA apart, essentially RNA is one strand of DNA, except just the U's and the T's are different, but they're essentially the same type of strand. So RNA can serve 2 functions. It can both keep its information, but then if it wants, it can just like take a strand of itself and fold itself up and start being a little molecular machine. So the belief is that early on, ribosomes were just, there was some spontaneous occurrence where all of a sudden you had this strand of RNA which was just kind of chilling there, and it happened to fold into the right configuration, and it just sort of red itself, and that was the beginning of life as we know it, which is, you know, a fairly rare occurrence, I would say.
So within the cell, what you're showing there is the creation of a protein.
Mhm.
How many of those are being made within a cell at any one time?
Oh, hundreds of thousands. It's a lot. I don't know exactly, but I mean, a tremendous, tremendous number.
Like in a second?
Yeah, yeah, yeah. There's just tons of these hanging around all the time, and on one single strand of RNA here, you might have 30 different ribosomes doing it all at the same time. They're just sort of following a leader and running around the RNA and just and different ones, so that I was initially saying that ribosomes get attracted to this conserved part of the RNA. Based on how sort of attractive that conserved part is, will dictate how many ribosomes will bind. So if you just want a protein to be formed in very low levels, you might only have one ribosome. If you look at it microscopically, you might only have one ribosome on a single RNA strand. But if you're needing a protein to be produced in huge numbers, so like actin, for example, is always produced in huge numbers, you might end up having 50 ribosomes on the RNA at one time. So it's...
Is that how you can tell under a microscope kind of what's being created?
No, so we don't actually, you can never tell that. The only way we can tell what protein is what is...
Testing it
By-
chemically?
Yeah, so we actually have to take what we know. So if we have to take its sort of match. So all these proteins will have something that binds to it, and that's the only way we can really know what it is.
So did the RNA actually preclude the nucleus?
Yep. Oh yeah, the RNA precluded everything. Well, except for the cell membrane.
So it created its own
Yeah.
sort of home to live in.
Mhm.
Where else does RNA exist?
It exists in every living thing. That's pretty much it. And then viruses. So any cell and any living thing will have RNA, but they also have DNA and then
Living,
virus.
including like plants and...
Yep.
Okay.
Yeah. So anything that you normally think of as having life has DNA in it. And then there are a few things that only have RNA. So there are some viruses that only have RNA. And that's why it's probably because viruses are very primitive and they're pretty stupid too. They don't have their own machinery. Or they're super smart because they know how to hide.
Right, I was
really
gonna
well.
say, I don't know,
Yeah.
they're stupid.
Yeah.
They're not little cells.
But I would say that I have a hard time believing, or I don't think of a virus as living. That's a really fun philosophical thing, conversation over beers, I suppose, whether or not a virus is alive.
They're not living or dead.
They're not living or dead, yeah, they live somewhere in the middle.
They just need to be.
Schrodinger's cat.
Yeah.
So is all this activity just to continue, like just to make more cells?
Yep.
That's it?
Well, it's just to make more cells, or if you look at the life of a human, I mean, it's not to do, all this activity is not to do anything, really. I mean, nothing in here, this is why I started out saying none of this is alive at the intracellular level. It's all just doing it because that's what that cell does. It's not actually thinking, it's just getting told what to do, and it's just based on chemical signatures.
It all seems so
So.
pointless.
How do we end up being able to do any of this?
Well, it's all just based on evolution. So you have two proteins. What makes that protein stick around longer? And it's because it maybe comes in contact with another protein, then it says, oh, well, it's not even trying to stick around longer. It's just that if it does happen to stick around longer, it has more of an opportunity to create another one. Or in the case of cells, it's the whole evolution is sort of pointless in a way. Evolution is sort of blind. It's not trying to evolve. It's just based purely on the fitness of whatever it is that's evolving. So you have two cells, and if this one ends up getting some compound or chemical or producing a mutation that creates a chemical that allows it to just live for 20 seconds longer on average than this one at the early parts of existence, then it's just going to have a higher opportunity to successfully create progeny, and this one's going to die out. And so that's the whole theory of evolution, is just nothing's trying to evolve. I mean, until, humans, we try to evolve ourselves, but we can't, well, I was gonna say we can't mutate ourselves, but that's not true anymore. The same thing at this level. It's just all based on what happens to be, if a protein gets mutated for whatever reason. So if this is whatever protein, if it just happens to get a random mutation, so I said that mRNA likes to be proofread before it leaves the nucleus. Sometimes that proofreading isn't perfect, and you'll get a mutation in the DNA or in the RNA, but evolution happens because of mutations in the DNA. But the proofreading isn't always spot on, just like in humans. And if you happen to get the right mutation that causes maybe this thing to latch onto this a much stronger fashion, maybe that will cause this protein to stick around longer, and that one cell that it was in will then be more likely to outlast its neighboring cells, things like that. So that's how evolution kind of proceeds. Okay, so this is kind of cool. So this is a ribosome. Again, that's the mRNA right here. This is the ribosome that's going to make the protein. It's going to make the string of amino acids out of the mRNA. But what it's doing here is it's actually going to, it's going onto the cell, the surface of the Golgi apparatus, or the endoplasmic reticulum, which are, we call it the ER a lot. It sort of just encapsulate the nucleus. And so what happens is the mRNA comes out, a ribosome latches on, but if it's a certain type of mRNA, and it will actually want to be formed inside of a vesicle. So instead of producing an amino acid chain that turns into a protein that's just kind of floating out in the cytoplasm, it will actually maybe want to, for whatever reason, say it wants to get out of the cell. If it's a chemical signal, a chemokine that needs to go and tell other cells, hey, we have this inflammatory process that we want to get started here. It needs to package them in a vesicle so that that vesicle can then bring it to the cell surface and let it out into the world. So what happens, in order to make a protein in order to put a protein into the vesicle, there's nothing that's going to allow that protein to just enter the vesicle after it's made. So instead, evolution's been really smart, and it tells ribosomes, hey, look, I made you a translocator. And just, when you go to make that amino acid chain, just kind of like poop it out right into the vesicle. And that's essentially what it looks like. And so you can see here, it starts to translate it, but instead of it going out into the cytoplasm, it's actually going straight into this vesicle, which we're just seeing the surface of a vesicle.
And nothing else can get through there?
Correct. Well, other things kind of can, but no, vesicles aren't made to like have transport in and out, right? They're just kind of sacs. That's what the little kinesin was carrying. And so they just, once they're in there, they're just kind of in there for a while. We use vesicles, our whole cell is made up of these little vesicles, and the vesicles are actually made up of lipid membranes. So I showed the cell membrane is made up of all those lipids. Inside of all the cells, you'll see all these other little vesicles, whether they're for killing microbes in a white blood cell or they're for transporting stuff. It's all made out of the exact same lipid membranes, which you'll see in a couple minutes. We want them all to be made out of the same thing because it enables them to all be recycled. So just like two bubbles, if you take two soap bubbles and bring them together, the reason that they can come together and then become one big one is because they're made out of the exact same material. So these vesicles, if we have the cell membrane here, and we have a vesicle containing a bunch of proteins that we want to get out, it floats up, floats up, floats up, and eventually once it gets to the top, just like a bubble might fuse with the other bubble, this thing fuses with the cell membrane, and when it does that, it involutes, and it lets out all of its contents outside of the cell, which is super cool. So that's being put into the vesicle. So this one is cool. This protein, what do you think this one is going to be? What kind of protein? Is that going to be one that just like leaves the cell at the end of the day? What happened to it? If this is a lipid membrane that we're seeing these waves on.
It's got those water on the end and lipid.
Exactly. So this is one they're showing here. It's going to become a transmembrane receptor later on. The cool thing is this part is on the outside of the vesicle right now. When that floats up and it involutes, it's then going to become on the inside of the cell membrane. So it's on the outside of the vesicle, but when the vesicle opens up and becomes one with the plasma membrane, what was once on the outside here becomes on the inside of the plasma membrane and remains as the inner part of the receptor. And then that part becomes on the outside of the cell. So this is what I was saying. This is essentially, the endoplasmic reticulum is considered, there's a very high concentration of ribosomes that are just all the mRNA. So the endoplasmic reticulum, if you look at the nucleus in the cell, it just is these amazing layers of plasma membrane that are just circling. If you look at a microscopic image, it's just like, it just looks like shale or like some liquid shale or layers that are just kind of circling around the nucleus. So the mRNA gets thrown out of those pores and just kind of goes immediately right near the endoplasmic reticulum. And the stuff that gets made here are those where the ribosomes are going to poop the amino acids into it to then create these vesicles. So now the endoplasmic reticulum is kind of full of these proteins that were translocated inside, and then it's going to just start budding. And it's not starting, it's always doing this, but it just buds off into these sort of vesicles, and each one of those is containing tons of proteins. And then this guy takes them and carries them to the cell surface, or they just float up. So, and you can see, so what they're trying to show here, these colors here, they're trying to show all these different proteins. So each one you're seeing here and here and here and here, those are all going to be transmembrane receptors.
And did they get there by floating up and attaching to that, or were they placed there?
These guys,
Mm-hmm.
that was where,
Is that what came out?
yeah, like came right out of the translocator there.
This is so
That was a
funny.
poop.
Yeah. So if you'll see, they'll either go all the way through right here, so this ribosome is making it into here. It will either go all the way through and then it will become actually an extracellular receptor, one that can go and signal to other cells. Or in this case, oh, it didn't show it, but it never finished going through, so the last little head of it was not able to pass through this translocator, and it ended up just kind of getting shoved out of the translocator and into the membrane. These little kinesins here, by the way, which is just another interesting thing. That's actually how our muscles work. So our muscles are made of all these different, it's made of a different type of tubule, but kinesins and dyneins. Dyneins are a very similar little walking thing, but they're more like arms. They grab on and go like that. Essentially, a muscle is just made out of these like striated layers. And each layer in between the two, and this is at the more molecular level than what you see. If you look at a muscle, you see striations. But at the very microscopic level, you also see these filaments. And it's made up of just tremendous numbers of filaments, and in between each of those is tons of these kinesins and dyneins. And so when you contract your muscle, essentially it's just all these little arms pulling each other in like that. And so it just contracts, and it just pulls the layers next to each other, and then when they relax, they start to come out again. So it's kind of this nice thing. But the reason that you get rigor mortis when you die is because you run out of ATP. So all the feet, they contract and then they can't relax again because there's no ATP anymore, so they just get stuck in that conformation.
Interesting.
The rigor mortis doesn't last, so what happens when it releases?
Then the proteins start to degrade.
Oh.
So all these things have half-lives. So this is a Golgi. So this works in close combination with the endoplasmic reticulum. So the ER will make these vesicles, but then the vesicles just start leaving the ER haphazardly, and they don't really know what to do. So the Golgi, and I don't know enough about this to know like how, and I don't know if anyone actually does, how the Golgi actually determines, but the Golgi is sort of like the central processing area, where the vesicles will move from the ER, they'll go to the Golgi, and then somehow the Golgi will then dictate, okay, you go this way, you go that way, you go to the cell surface, you go to the nucleus. And it's sort of the real processing center of the cell. And it's just made up of all these little layers. And so you can see the vesicles are binding, they're gonna let their proteins into the Golgi, and then the Golgi is gonna take up all of them, just like a letter, just like a mailman delivering to the post office.
Does it organize them?
Yeah, so that's exactly what we're seeing here is, you can see the vesicles on the bottom that are floating up, they're floating up with a bunch of protein coming from the ER. Those vesicles will fuse with the Golgi, it will let the protein out, and then the Golgi actually collects all the proteins and says, okay, you're all this type of protein, you go that way, you're all that, you go that way. And that's essentially what's happening with these. These have now been sorted and they're ready to go. Some of them will just float to the surface and some of them will be carried. Like this.
So that's a protein, not a vesicle?
No, that was a vesicle being carried by one of those kinesin things. So that big, it was just like a big sac. So this one, what we're seeing here, it's too bad they didn't show it actually, but this is, they're showing the bottom side, or they're showing the outside of the cell now. The vesicle has just fused with the plasma membrane, and so it created this little involution, and so it's letting those proteins that were put in packaged inside the vesicle, it's letting them out into the extracellular
Is
space.
the Golgi inside the vesicle?
What's that?
The Golgi's inside the vesicle?
No, the Golgi is what becomes the vesicle essentially. So if you think of the Golgi as more plasma membrane, the vesicles will fuse with the Golgi. They'll let their internal stuff into the Golgi, which is just kind of itself this huge sac, just like a really flat, long vesicle. And then from the Golgi, you'll have more of these little round vesicles that pop off of it. And then they'll go from the Golgi apparatus straight up into the plasma membrane. Once they fuse, they'll allow their internal stuff to leave. And so now we're sort of looking outside. But it's all made up of plasma membranes in that case. And then you see the ones that kind of just stick on. So these used to be inside of the vesicle, and now they're on the outside of the cell.
So are we looking outside
Now
the
we're
cell?
outside of the cell again, yep.
Okay.
This is just redoing it. So now you can see cholesterol coming together to make a lipid raft. And that allows the integrins to kind of stay still and they create these junctions.
So cholesterol actually has a really important role.
Oh, we would die if we didn't have cholesterol. We would die immediately.
Okay, because you said,
It doesn't have a role for, at least not that I know of, it doesn't have an evolutionary role in terms of like creating the white streaks, and it just kind of builds up
but it
by
has a huge
mistake.
role in the life of the cell.
Exactly.
Ah.
So this is really
So
cool. I
what
love
does it
this.
actually do?
Yeah.
We saw those microtubules and actin filaments, how they can easily build themselves up and then break themselves down. So cells need that. I mean, we would love it if we could like, just in an instant be able to like squeeze under the door or something. But cells can, and they actually like can just break down all of their cytoskeletons. They break them down based on chemical signals that say, okay, you're where you need to go, the chemical gradient has gotten enough. We need you at a wound right outside of the blood vessel here. So they kind of roll and roll and roll until the chemical gradient gets strong enough. And then they start to just like break down the cytoskeleton until they're like these little pancakes, and they can then just squeeze through the endothelial layer, which we need because we wouldn't want the endothelial layers, the blood vessels, to actually come apart so much that a round cell could move through, because then we'd end up getting edema and we'd die from exsanguination very rapidly. Instead, they have this amazing ability to just change their shape to work with us, so that they can just kind of squeeze through the blood vessel layers and out into the tissue where your infection is or where your cut is. And these are the things that create pus and stuff when you get cut.
And so that's a vesicle.
No, this is a leukocyte, a white blood cell.
Oh.
So that was one thing that we just spent the whole video inside one of these.
So this is all about life, this is not pathological,
What's that?
this is kind of not, not pathological.
Right, this is just about how life is working.
Okay,
And
that's
they're
it.
using a leukocyte as
It's
an example.
exiting this.
It was exiting the blood vessel right there. And the reason it was doing that, so truly if I get cut here, somehow my blood cells need to know, okay, I'm cut here or I have an infection here. And it needs to know, it needs to get out into the tissue somehow and they're kind of stored in our blood vessels essentially. So how do they get out? They just kind of follow the trail and then they squeeze through and get out there. Which is really, I think it's super neat.
There's so many great implications for nutrition.
Yeah. I mean, it's in terms of everything, it's just the life of the cell is pretty damn cool.
That was great.
Do you want to take a break for a minute? And
Yeah, bathroom break would be good.
Good countdown.
So, let's see. I could keep talking about cell biology, but I would like to, I think, see if you guys have questions about what we just saw. Because, I mean, this stuff, we could talk about this for 2 years and not finish up. Because I guess what I'm wondering from you all is if you would prefer to keep hearing about this stuff or talk a little bit about a whole different thing, which is sort of this vaccine, antibiotic kind of thing, just because there's such conversation in the public
And
about that right
mutations.
now. Sorry?
Mutations?
Well, we could talk about mutations. That ends up being a whole different thing, but I'm happy to talk about that too. Because it has to do with antibiotic resistance anyway, and evolution in that
I'm
regard.
taking antibiotic right now.
And you're probably creating bacteria with little mutations too.
Right, exactly. For Lyme disease.
Oh yeah, yeah. So
What did you say, I'm sorry?
Lyme disease,
Oh.
yeah.
being treated for that.
The videos that we have lined up that we've shown in the past are pretty quick.
Yeah, I can show you
We want
this.
to
So
just show them just for the sake of
yes.
the bigger picture of cell division and then move on.
Yeah, so I'll show you this one. So mitosis is the process of cell division. It's how babies are not born but formed, and how all of our cells divide. So what happens is we have these chromosomes that hang around in the nucleus. The chromosomes have to then come apart, and we have 2 of each chromosome, which is important because then they can each go to a daughter cell. And so I think I don't want to talk about mitosis too much unless you have particular questions, as much as just show you how it looks so that you can see how amazing it is. So this is a nucleus we're looking at here. Let me make this bigger. We're looking at the nucleus of a single cell. So it's zoomed way in. And you'll see the nuclear membrane, which again is that lipid bilayer, break down, and you'll see the chromosomes start to condense and pull apart. So there, this video really has a hard time showing this part of it. But essentially what just happened, normally we don't see, these are all chromosomes, so these are just DNA right here. When the DNA is being used in a cell to produce proteins and things like that, it's going to be just like spaghetti. It will be all sort of out and loose, and that's to enable the transcription proteins to actually bind to the DNA and transcribe whatever pieces of the DNA it needs to create a protein. But when a cell wants to divide, all of that DNA will actually condense into what you probably think of as a chromosome if you've had any sort of cell biology in the past. It's these sort of X's, you see X's, or in the case of the male, the Y. But they all kind of look like this sort of shape. So that's just condensed DNA. And that's what we're seeing here. This is the condensed DNA. And what you're seeing is it's forming on top of, at a plate here. And it forms right before it needs to get pulled apart. So now we have a full complement of each of the chromosomes on either side here, here, and here. And then you'll have those, earlier I said that there are those centrosomes which end up like pulling the DNA. We saw a brief picture of it. What they do, it's quite amazing. Actually, the next video will show it. They send off little spindles and they actually latch onto the chromosomes and pull them to either side of the cell. And so that was the chromosomes actually getting pulled apart before it produces a daughter cell. And now you'll see the nuclear membrane reform around it. So that now it's got this whole, a lipid bilayer just reformed around this. So it just became a little sphere instead of a sphere. And now it's pinching off and creates two daughter cells. So that was just a real... Do you ever notice that was from Lehigh?
No, I just noticed it for the first
Yeah.
time.
That's funny. So here's a more animated version which will show it a little bit better. I think there's maybe some sound here.
This is a really silly tune, I think. Oh no,
Oh,
this isn't the one. This isn't the fun one.
So here, this is again showing inside of a cell. This is like a strange godlike picture. This is inside of the cell, so this is again the nucleus. And they're not showing all of the chromosomes, it's just for visualization, but these might be like I think they're just color coding them just to show which ones are going to which. Here they're showing the centrosome here, and I don't know why they're making it look like a sun. But you'll see the chromosomes, so you have these two centrioles. They'll kind of go around to each side, and they send out these little spindles. They cause this membrane here, it will start to break down. Now you have no more nucleus, and you just kind of have free DNA sticking around in there. These chromosomes line up, and then in an amazing fashion, the centrosomes send out, these are microtubules, which were what the little things were walking on before. I said that they're also kind of like the ropes of a cell. They do a lot of the lifting and the pulling of a cell and things like that. So these microtubules they just start building. So they're actually getting lengthened as they're moving. They're not just like stretching out. They're actually have those little molecules building them up as they're moving along. And they get built up straight into one of these. So this is the centromere of I can't remember what this little part is, but this is one chromosome here. And they bind to those two little parts, and they'll just pull it straight apart. The way that they do that, there's actually this process where microtubules start to contract by disintegrating from the inside out. So they're actually disintegrating, and that pulls them this way.
And over what period of time is this happening? Or
This is all happening within like an hour or so. Less than an hour, actually. This part. So now we have the nucleus sort of reform around the chromosomes, and then the whole cell is going to divide here. So this is sort of how all cell division occurs. Well, it's not sort of, that is more or less how cell division occurs. And now we can watch this. This is a real life version.
wait, this is on we saw before.
Yeah, no, I'm just playing it again that I've seen.
Oh,
Do they call them daughters because they both have the X's? Or
What?
daughter cells, not son cells?
I was wondering about that too. Why they call them daughter cells?
Because they
I don't know.
Well, who gives birth?
Oh,
the
yeah,
daughters?
right. Yeah.
So they have to grow up and give birth again.
Ah.
I feel like, but we also, in scientific literature, we just tend to always call things daughters. You never hear the word son be used for anything.
Or offspring, or you could use like a number of other.
Yeah, but I mean, for cells, we would, for some reason, we always tend to, for things that divide, we just always call them daughter this and daughter that, which is kind of interesting. I never really thought about it, but I would think that it's because they're the ones that actually.
You're like Dura Mater, tough mother.
So here, this is super cool. This is actually what happens with the immune system. And pieces to it. And the neutrophils just chasing it, and that's exactly what happens in our body. all
But
the
why
time.
does it it's not gonna eat it, what's it gonna do with it?
It does, well, it eats it in terms of it engulfs
It
it
absorbs it.
and it puts it into vesicles actually. So the way that it engulfs it is, I wish that other video was showing vesicles going to the cell surface and letting stuff out. They also, with clathrin, well, there's a lot of different ways to do it, but they can actually engulf and create new vesicles. So the way that our immune systems kill stuff, by and large, is by engulfing stuff. So it will actually grab it, it will pull it in, it will wrap its membrane around it, and then kind of create a new vesicle inside of the cell. And that vesicle now contains the bacteria, but these are very specialized vesicles. They end up having little, tunnels and stuff that allow acids to come in, so it ends up making a very acidic environment inside of what's called an endosome and a lysosome. So the endosome is what takes the bacteria in. So the neutrophil creates this specialized vesicle, captures the bacteria, pulls it into a vesicle. That endosome then fuses with a new vesicle called a lysosome. Those fuse, and the lysosome has a very low pH, so it's very acidic, and it kills the bacteria. And that's how we kill stuff in our bodies.
But isn't that also how HIV counteracts that, is it breaks down the walls of the cell so it can't engulf it?
HIV is a lot of different things, but it does prevent endosome, it prevents endosomal lysosomal fusion. It also just starts killing CD4. HIV's major thing is to just kill CD4 cells, which are required to maintain the adaptive immune response. So as you start to get low enough numbers of CD4 cells, your immune
That's where you get the
memory
ACE,
just starts.
yeah, that's where you get the ACE
Then you
diagnosis
get
based on the
exactly.
low.
Opportunistic infections,
Yeah.
which really do you in any way.
Yeah, HIV won't ever kill you. It's
Mhm.
just
The medications will before the,
Yeah. So here, we'll play this again just so you can appreciate how cool we are.
Benny Hill. It's before that. No, it's,
It's been a month. Unless it was before that too.
yeah.
Right there, it ate it. I don't know
Yeah.
if you guys saw it.
Yeah.
Just
Yeah.
like encapsulated at the very end.
What were the other little circular things in there?
Let's see.
Were they just,
Oh, these? These
Yeah.
are lymphocytes.
ah,
They're either lymphocytes or they could also be just bubbles. I mean, depending on what this is. But if this is actually a blood sample, these are probably lymphocytes. These are the lymphocytes are part of the adaptive immune response that vaccines and immune memory are used for, or use. And then this is part of the innate immune response.
what's around them? It looks like they're glowing, I know it's not glowing,
This?
but what is around them that?
Oh, this
makes
is just because they're round, and this is a light microscope, so the light is shining straight through. So it's just a dark field around it.
so this is within one cell?
No, each of these is a cell in a petri
Okay.
dish. So this is if we took a blood sample and threw it on a petri dish. Now we're seeing some white blood cells. These are lymphocyte white blood cells. This is neutrophil white blood cell, and then these are bacterial
And
cells.
what is the substance that they're in? Is that cytoplasm? Or what
No,
is-
cytoplasm
what are
is
they in?
inside of each of these cells.
So what's between the cells?
They're in a petri dish.
They're in a petri dish, so saline could be anything.
Okay,
Could be if it's blood, then it's just the plasma. from the
and in
blood.
our bodies?
In our body, it would be plasma, or if it's if we're talking about in the blood vessels. Otherwise, it could be.
Tissue.
Yeah, it could be interstitial fluids and stuff
Mhm.
that exist between all the tissues and things like that. Which is just salt water, more or less, with a lot of other stuff in it. Okay, so that's our sort of entrance into immunity, I suppose. You guys have any questions about anything thus far? So we'll kind of take a very different direction once we start talking about vaccines and things like that. And I don't have good pictures for vaccines, so I'm just going to have a conversation about them, I think.
I have a flip chart here.
Can I ask you, like, in the sensitivity when you have